We're going to get right into our first session today. So, as provinces across Canada work to advance access to comprehensive genomic profiling, clinical teams are navigating varying system conditions, infrastructure, and expectations. This national roundtable brings together frontline clinicians to build clinical consensus about the utility of CGP as a standard of care and to identify the conditions needed to support equitable, informed clinical decision-making across Canada. This discussion will be moderated by Dr. Monika Krzyzanowska, Chief and Medical Oncologist at the Odette Cancer Centre, Regional Vice President at Ontario Health, as well as co-chair of the Expert Steering Committee and a valued member of CRAN's Medical Advisory Board. We also welcome Neil Marr, patient partner with PanCure RX, who represents the caregiver perspective. And a warm welcome to our clinician roundtable, which includes Dr. Meeta Manna, medical oncologist at Saskatoon Cancer Centre, Dr. Stephanie Snow, medical oncologist at QE2 Health Sciences Centre and valued member of CRAN's Medical Advisory Board, Dr. Rebecca Auer, Executive Vice President of Research and Innovation at the Ottawa Hospital and CEO and Scientific Director of the Ottawa Hospital Research Institute, Dr. Shantanu Banerjee, co-chair of the Expert Steering Committee, medical oncologist and director of Precision Oncology and Advanced Therapeutics at Cancer Care Manitoba, Dr. Yuj Ko, medical oncologist at St. Michael's Hospital Unity Health and another valued member of CRAN's Medical Advisory Board. We have Dr. Urban Emenegger, medical oncologist at the Odette Cancer Centre, Sunnybrook Health Sciences, and Dr. Jason Karamchandani, president of the Canadian Association of Pathologists, and yet another member of CRAN's Medical Advisory Board. This is quite an esteemed roundtable and will undoubtedly be a great discussion. The roundtable will proceed until about 11:15 Eastern Time, at which time we'll move to Q&A. So please do feel welcome to submit your questions and comments throughout the discussion using the questions bar below the video stream. We look forward to getting to them. And Dr. Krzyzanowska, I'll hand it over to you.
So thank you so much, Cassandra, and good morning, everyone. It's a pleasure to welcome you to this session, this National Clinical Roundtable on the Comprehensive Genomic Profiling, or CGP. I'd like to thank Michelle, actually, for her words. It was a nice opening, sort of right prior to our session. I was listening to that. So thank you, Michelle, for sharing your story and for all the work that you do to help support patients and support care for patients with cancer across the country. So today what we're going to talk about is about the evolving role of CGP in clinical care across Canada. As many of you know, cancer care is increasingly being shaped by our ability to understand the biology of an individual tumor, and Michelle talked about that actually in a lot of detail in her story. Biomarkers play a role in different parts of the journey, in diagnosis, prognosis, treatment selection, and especially in that treatment selection piece, selecting drugs that may or may not work actually for a particular patient. So, as a medical oncologist, this is the type of information that I utilize on a regular basis to help me identify therapies for my patients, or identify clinical trials, which is an important consideration, or other treatment pathways that may not be apparent from histology alone. And I know over the last day or so, you've already been talking about CGP, so I'm not going to get into a lot of details about that. But really, what we're discussing today is why CGP versus the current approach, which is often either a single test or a group of tests for a particular cancer. And from a clinician perspective, I think what this can help us is really provide a more complete picture of what's going on with that particular patient's cancer, especially in the advanced disease setting, and then help us make decisions in a more timely fashion where timing is important and treatment decision-making is quite complex. But at the same time, the value of CGP doesn't depend just on the test itself, but also on the systems around it. And so that's what we're going to talk about today: access, eligibility, turnaround times, things like that. And we have a great panel here from across Canada to talk to talk about from different perspectives as well, not just different geographic perspectives, but also different clinical perspectives. We have medical oncologists, surgical oncologists, pathologists. So really an excellent panel. So across Canada, what we've seen is that provinces are moving under different system conditions and with different infrastructure, resources, and pathways. So the goal of this session is not really to prescribe a single type of an approach, but rather to ask where greater clinical alignment is needed and what conditions would help support equitable, informed decision-making for patients and healthcare providers, regardless of where they live or practice. So we have three main objectives of today's discussion. Those are to examine the clinical value of CGP relative to current testing approaches and how it can support more informed treatment decision making. Second, we'll talk about some key clinical, technical, and system conditions that are needed to integrate CGP effectively and sustainably into routine cancer care. And third, we will explore where there may be opportunities to strengthen consistency in the use of CGP across clinical practice, care settings, specialties, and jurisdictions. So as I said, we're very fortunate, Philomena has already started with that, to have a very thoughtful and diverse panel with perspectives from across disease sites, country, clinical specialties, and importantly, we have the caregiver perspective included with Mr. Neil Marr joining us for this session. So as we move through the roundtable, I would encourage all of us to think not only where CGP adds value today, but also where evidence and actionability is still evolving. What minimum conditions are needed for CGP to be useful in real-world clinical practice, and where variation in access or implementation creates the greatest challenges for patients and clinicians. And so with that, I would like to invite Neil to begin by reflecting on his wife's cancer journey and what access to CGP as a standard of care might have meant for her care and their treatment decision-making. So Neil, thank you for being with us this morning and for sharing your story and your wife's story with us.
Well, thank you. And hello to everyone in the meeting. First of all, I appreciate the fact that we have conferences now with patient caregiver perspectives. I think it's so important because while I may not be able to speak to the scientific aspects of this very clearly, I can offer another dimension, which I do think is important. By way of context, my wife was diagnosed with cholangiocarcinoma in 2019. And it's a little unusual in that most people, when they're diagnosed with this, probably haven't even heard of it. It's a rare cancer, but in fact, her mother had been diagnosed with this and had passed from this in 2010. So we were quite aware of the gravity of the situation. But as I reflect on the journey with profiling, it was through my own research that I discovered this was even a thing. And by 2020, post-surgery, I asked the oncologist about it. And she said that we don't do this yet. And of course, 2020, we're in COVID. And sometimes an outsider perspective, whether scientific or not, can be helpful, because when you're in a culture that's the way things are done, you sort of accept those things. I sometimes reflect on, I came to this country in 1974 as a child, and I remember halfway across the Atlantic looking around at all these people smoking and thinking the nearest fire station is in Reykjavik. As a child, I had an outside perspective, and as a caregiver patient, I have an outside perspective. And the first thing I thought, well, why not? Why would we not do this? So with some probing, discovered that we could get the profiling done at Foundation One at considerable cost, which was obviously not an issue to us. It was important. But if I could just boil it down to one thing, when you're going through a serious diagnosis and disease like cholangiocarcinoma, and the prospects can be daunting, hope is such an important factor. So as I said, I'm not speaking to the science, I'm speaking to the hope factor. And this gave us immense hope and kept that going, which is important from my perspective as a caregiver, to have that. So it was less stressful. Now, the advantage, we've come a long way, I think, even in these few years. But the advantage, I would say, of getting it done early, which was what I was pushing for, is because sometimes mistakes happen, and particularly during COVID. In fact, when the specimen was sent, it was first lost, and so there was a delay there. Now, as it transpired in our situation, there were no actionable mutations, but it's a step that was so important that we would have, not just because it may have offered, because some things were coming online, but again, it just gave us that hope to keep us going.
Thank you so much for sharing that. And, I like that. Why not, right? That's sometimes that's an important consideration as well. And the importance of hope as patients are facing these difficult diagnoses. So thank you for sharing your wife's story, Neil. So maybe I'll kick us off with the first question for the roundtable, and maybe we'll kind of go to the basics and talk about the clinical utility of genomic profiling, comprehensive genomic profiling. So the question for the panel, and I'll maybe start off with Dr. Karamchandani, and then a few of our medical and surgical oncology colleagues thereafter. Where does CGP add the most clinical value today, and where is their evidence or actionability still evolving? So, Dr. Karamchandani.
Monika, thank you very kindly. And I can say it's a joy to be here, so thank you to CRAN for the opportunity. And I'm here invigorated in part because less than 2 weeks ago, Dr. Snow presented in Prince Edward Island and presented data of drugs that are coming down the pipeline that are potentially truly game-changing in terms of prolonged and durable responses to cancers which in my lifetime were incurable. Now, for the patients and caregivers, I do want to give you a little bit of perspective. For a long time, cancer care proceeded as follows. A clinician would identify a possible lesion and have some index of suspicion that this required a biopsy or a fine needle aspiration or something to confirm a diagnosis of cancer. That went to my kind of doctor. That went to the lab in pathology. And on the basis of cytology, a biopsy or a surgical resection, a diagnosis of cancer was established. Now, we were, for many years, sort of troglodytes in that we organized cancer by the site of the tumor. So we felt we were doing a good job by describing something as prostate cancer versus breast cancer versus colon cancer. Mercifully, even over my career, the progress in therapies have forced an evolution in how we classify cancer. So understanding the site of origin is no longer enough. We need to understand the biology and molecular drivers of disease. And I'll go one step further to say, even understanding some of the targets that help my clinical colleagues determine therapies to guide patient management is now essential standard of care in 2026. Now, one thing I do think is challenging for patients and caregivers is even the word biomarker. Biomarkers can mean different things to different clinicians who take care of different kinds of cancer. So a biomarker could be anything from looking at a protein expression, and some patients will have heard about HER2 in breast cancer or PD-L1 in lung cancer. And there are new biomarkers coming out in ovarian cancer, in upper GI cancers that are protein-based. On the other side, we have the idea of comprehensive genomic profiling. And there we're looking at often mutations, amplifications, and translocations, which are the three main categories of genetic alterations that are known to participate in cancer. And all of these can be targeted. In fact, as I said, Dr. Snow's data on the second-generation ALK inhibitors have put wind in my sails as someone who helps diagnose and take care of patients with metastatic lung cancer, particularly in young women who are non-smokers. So here it is. It's 2026. In my own career, from the time of my fellowship, there were brainstem tumors which were incurable, but low-grade brainstem tumors. By 2015, we had a precision biomarker, which was based around immunohistochemistry, but looked for a targetable mutation. So my young patient in 2015, who was 7 years old, was placed on precision therapy within 5 days of a brainstem biopsy. In 2026, that same patient would no longer need that physical biopsy. We would do a liquid biopsy. We would do a lumbar puncture to look for specifically the same mutation we identified back in 2015 using tissue. Since that patient started therapy, the therapies have evolved. So the patient is no longer on the first line BRAF inhibitor. The patient is now on a combination BRAF inhibitor with fewer side effects and a MEK inhibitor. And so my patient from 2015, who was 7 years old with what was an incurable brain tumor in 2010, is now an 18-year-old man. So there is tremendous progress around us. And there are many ways that we can test for the different kinds of things we need to help guide patient care. And comprehensive genomic profile is no longer an aspiration for many Canadian patients and pathologists who like to order these tests to help inform our clinical colleagues, but it's increasingly becoming a reality. And the progress, even in underserved areas, particularly the progress I heard about in the Maritimes, is inspiring. So we're making progress. It's slower than it needs to be. And that's why we look to Cran and Philomena and this team to help us advocate to improve the rapidity with which we make this standard of care.
Thank you so much. Thank you so much for those comments and explanations. And indeed, it's a very exciting time in oncology with all the new targets and opportunities and why this is becoming even more and more relevant. So maybe we'll switch gears and get a perspective from some of our medical oncology colleagues and then from Dr. Auer. So Dr. Emenegger, what are your thoughts around CGP and where it adds more clinical value today?
So I'm a prostate cancer oncologist. I see primarily patients with metastatic prostate cancer, which is unfortunately still an incurable condition. And we do genetic testing since around the time when the pandemic started, but it's not comprehensive. So to give you an idea about the impact genetic testing has these days is that there's a group of drugs called PARP inhibitors, and these are drugs that typically do not work in prostate cancer unless these cancers have a certain genetic profile. And this profile is existing in around 10 to 15% of patients. So in 10 to 15% of my patients, doing the testing actually has an impact on giving them an additional line of therapy, as we have heard before from one of the patient representatives. There's another group of treatments that are widely used in certain cancer types that do not work really well in prostate cancer, which is immunotherapy. And only around 2 to 3% of my patients actually are candidates for immunotherapy, and we have to identify them. Now, the issue is, and this is why I say that we don't do that comprehensive, we started testing for these PARP inhibitors in 2020, 2021, and this has been established. There definitely are some differences across the country when it comes to access to the testing, but we do that now as a regular testing procedure in most of our patients. The issue with immunotherapy is that when the testing was introduced for these PARP inhibitors, we didn't introduce at the same time the testing for immunotherapy. What this means is that you have to go back again to get the samples. You have to retest the samples. So in essence, we just repeat things a second time. And this is where I see the benefit of being bold and actually being comprehensive from the get-go, because then you don't have to go back. You have the profile of the patient. And from the beginning, when you do the testing, you have all the information that actually is helpful for really providing patients with the best treatment options that they are around. And then maybe the last thing I want to mention from my perspective is that, and this is alluding to something that Jason actually referred to before, we also have a test called PSA PET scan. In essence, a PSA PET scan is a way to know whether prostate cancer cells express a protein called PSMA, which stands for prostate-specific membrane antigen, at their cell surface. And we know that this happens in around 85% of patients. Since the treatment that we can use is restricted to a certain stage of prostate cancer, this doesn't affect 85% of all my patients, but overall it affects around 25% of patients with prostate cancer. So when you add this together, around 1/3 of my patients actually are treated differently now compared to 5 years ago, because we have all this testing done. And with comprehensive genetic profiling, I would hope that these numbers will drastically go up, but we are still quite far away, at least in my field, when it comes to comprehensive testing.
Thanks for your perspective. And I should mention that Dr. Emeniger is in Ontario and Dr. Karim-Chamdani is in Quebec. So maybe at one point we'll talk about the differences between the provinces in how the different provinces are achieving or not achieving access to comprehensive genomic testing and to biomarker testing in general. So maybe Dr. Ko, do you want to chat about your experience? So we'll talk about Ontario, and then maybe I'll ask Dr. Manna about Saskatchewan before coming back to Dr. Auer and talking from a surgical oncologist perspective.
Thanks, Monika. And I appreciate the invitation to be part of this. And it's very timely. And I'm located in, I guess, the closest thing to downtown Toronto as possible, next to the Eaton Centre. And my clinical practice is focused on GI cancer, patients with GI cancers, mostly colorectal and hepatobiliary, and including pancreas cancers. And I think the word that has been used multiple times so far today is the word comprehensive. And I think that definition is changing to be more inclusive every year. And I think that will hopefully mean better outcomes for patients. This is a technology that was pretty narrow in scope just a handful of years ago, as the number of targets and available drugs was limited, but it continues to grow quite significantly. And probably this year marks the biggest advancement in the area of pancreas cancer and the potential targets. For example, although comprehensive genomic profiling was available for pancreas cancer in Ontario for a little while now, more than 90% of patients have a mutation in RAS in pancreas cancer, but this was not really a druggable target until this year, and it's not a home run, but it's certainly a good 6-0, such as how Canada did in the World Cup yesterday. It's a very good outcome. So I think CGP is having a better role or a more expansive role for all cancers, certainly in GI cancers, starting from colorectal to pancreas now. There are other targets that are being identified, such as NRG fusions, which are rare, but it's something that has to be identified for a drug to be used. And I think as this is done more frequently and more available for patients, ultimately the outcomes will be better. It's getting cheaper. It's getting more accessible. And one thing that has not been mentioned yet, but been alluded to, is the repeat testing that we had done previously for sort of one protein, one gene at a time, which spends down the available tumor tissue in patients. So doing it all at once, I think the word comprehensive is even more important then. Is really key. And I think there are many reasons to do this. And I think others have done work at looking at the economics of doing CGP versus multiple single tests in terms of the clinical relevance expediency should not be underestimated. So I'll leave it there. Thank you.
Thanks, Dr. Cohen. And maybe later, and depending how we can maybe come back to this idea, what does comprehensive even mean? With, there's been evolution in the testing and evolution in patients are living longer and when and all of that. So we'll come back to talk about the comprehensive piece. But Dr. Manna, maybe can I ask you about the Saskatchewan perspective or as a medical oncologist perspective, or both?
Absolutely. Thank you for having me here. And thanks to Cran for inviting me to speak yet again. This is, I think, my third year participating in this conference. And so I treat predominantly breast cancer, also some lung cancer as well. And I can say just during my short time period with Cran, I've seen a rapid evolution and change in how we approach and treat patients. So when it comes to breast cancer, and I think some of my colleagues have alluded here, what exactly is a biomarker? And it was historically one of the first malignancies where we sort of identified biomarkers, estrogen receptors, progesterone receptors, HER2, for example. And now we've seen an evolution where we're looking at various different mutations, whether it's hereditary or germline, or something that's acquired. And we now have companion targeted drugs that we're able to utilize. So we're not just looking at the original biomarkers, ERPR and HER2, but we're also, delving a little bit deeper. And even in the recent years, when we're looking at sort of how we're testing in each province, initially we would have to, if we wanted a certain biomarker, for example, PDL1 for triple negative, we would have to send it out of province or even out to the States to get it centrally done. And it would take, 10 to 12 business days for us to get those results back. And once again, there's always a concern, do you have enough tissue, et cetera? Now we can do it, locally and in-house, but we still have to request it. So it's not like we're getting a full panel, we're pinpointing which test we want. So you can see these changes are happening where initially, if you want a particular test, you might have to send it out. And then now we're starting to develop technologies where we're able to do it locally. And just as mentioned, we're trying to keep up with the rapid changing landscape where our pathology labs are trying to get better machines, better tests, better panels. And even I was just recently talking to one of our pathologists and talking about how we're testing for hereditary BRCA mutations. They have now notified me that they now have a nicer and bigger machine that can actually run faster panels, and now they can actually test for more mutations. So the panels are becoming larger. So things are changing and evolving with time. And I think groups like this help us identify sort of the need and the gaps, because we know that it's always going to take time for our public system to keep up with things and be able to test for patients appropriately. So when it comes to breast cancer, we know that we test for both hereditary mutations and acquired mutations. And as mentioned, it's nice to have a panel right up front to know sort of what is the patient's trajectory, what are some actionable mutations that we can target. But the other area in breast cancer that's also been evolving is instead of looking at disease progression on imaging and knowing, okay, this is when we change therapies, also looking at an entity where the molecular entity is changing. So looking at molecular progression. So not just at one point of time, but looking at a certain mutation called ESR1 and seeing when does that emerge and when do we then switch therapy. So that is sort of a novel concept that came out from one of the breast cancer studies. And so that kind of begs the question in terms of when do we test for biomarkers or comprehensive genomic profiling? When do we test? Is it upfront? Is it at time of disease progression? Which biomarkers do we need to test for? And do we need to test for all of them every single time, right? So I think these are some important caveats because we need to be sort of prudent in terms of how we allocate our resources appropriately, right? So we need to know what is valuable, how is this going to impact the clinician's decision, and how is this also going to impact the treatment for the patient, right? So I think these are all important questions when we're looking at CGP and various biomarkers. And as I said, in Saskatchewan, this is what I've seen kind of evolve in our province, but working also collaborating with other centers and other colleagues across the country, I think we're all facing similar barriers in terms of how we can ensure that our centers are testing patients timely, equitably, and making sure that, once again, no matter where you reside or where you're being treated, you're getting timely access for testing as well as treatment.
Thank you, Mita. I think those are very important points because I think, what do we mean by comprehensive? And Michelle talked about, I think, some of her experience with the ESR1 and the, the need for repeat testing. So how do we make sense of all of that? It's a very complex and evolving field, and then the technology is advancing as well. So we'll probably have to come back maybe at the end and talk about what do we prioritize in terms of where would we need the most alignment. But before we talk about maybe about the system level things, I did want to come back to Dr. Auer and get her perspective on this topic about clinical value and of comprehensive genomic profiling from a surgical oncologist, clinician scientist type perspective. So, Rebecca.
Thank you. And thank you so much for the opportunity to participate in this panel and for all the work that CCRAN does in this space. I think it's so critical. I'm kind of speaking from the perspective of rare cancer. I founded the Canadian Cholangiocarcinoma Collaborative, and as many people know, biliary tract cancer is a rare cancer, but it's also one of the most molecularly rich in terms of targetable mutations. And so it creates a series of ultra-rare cancers, and that presents its own challenges. But I think those challenges are just highlighting the challenges that all cancers, whether they're common or uncommon, have. And I would say we fundamentally need to reflect on two questions that were kind of already touched on today. One is, what do we mean by standard of care? And what do we mean by comprehensive? And I think standard of care traditionally is fairly narrowly defined. So if you have a drug that is targeting a very specific mutation, and that drug is Health Canada approved in breast cancer, then that molecular profile for that specific mutation would then be standard of care for that cancer only. But I think that means that we will always be flat-footed. Every time a new drug becomes approved, every time we have an exciting ASCO presentation and there's a game changer in the space, it takes us months to years to kind of be able to figure out how to implement that specific profile, that specific genomic or biomarker test within our various pathways. And I think if we start to say, well, standard of care is expanded, standard of care also includes an opportunity to participate in research, in clinical trials, an opportunity to try an off-label drug, particularly for rare cancers, then we change when those biomarkers start to be integrated in that pathway. And this makes a lot of sense, as the current oncology paper from last year really highlighted. Larger panels there's a better return on investment per gene if you do a larger, more comprehensive panel. And so, and those panels are getting cheaper and cheaper. And so, if we think about, let's say, just if we don't focus on the protein space, but just on the NGS or the genomic space, remember that it took 13 years and $3 billion to sequence the first human genome just over 20 years ago. And the pace at which we've been able to do this now is incredible, and it continues to get cheaper. It continues to get more accessible as we become more creative with our technology. So I think we should be thinking about standard of care as anything that is on the horizon where a clinical trial is available, or where there is something approved, maybe for a common cancer, a different indication, but there might be an opportunity to use the drug on an experimental protocol or an off-label protocol. And this is particularly important for a rare cancer like cholangiocarcinoma. We have many patients who will have a mutation, let's say BRCA. We just talked about BRCA mutations in breast and prostate, and PARP inhibitors are approved in those indications. There's probably 50 or so patients in Canada with BRCA-mutated cholangiocarcinoma. There will never be a clinical trial or a Health Canada market authorization. Does that mean those patients should never access a drug that could be highly effective? So I think we need to think differently about standard of care. Standard of care includes opportunities to participate in research. And comprehensive, I think we need to think differently about that as well, for the same reason. We shouldn't be playing catch up every time a new marker comes in. We should be forward thinking and future proofing our processes. And that includes education all the way from the very first biopsy that's done. It can't just be, one pass of a core needle to make a diagnosis. This is a breast cancer. It has to be 6 passes of a core needle, so we have tissue available for future opportunities for clinical trials, for molecular testing, for new biomarkers, and not, as someone else pointed out earlier, not requiring patients to go back for another biopsy or another test because there's insufficient tissue to do another biomarker. So I really think we need to integrate this concept of research as part of care. Research is something we need to have a responsibility to offer it to our patients. And in the only way we can do that in the cancer space, in my mind, is to have a much more broad remit on biomarker testing.
Thanks so much. I couldn't agree with you more. I used to treat a rare cancer, thyroid cancer, and we had an opportunity to have patients participate in comprehensive genomic profiling as part of a trial at a time when actually we didn't have any drugs that targeted any of those mutations. But as we collected the data, the drugs came out, BRAF inhibitors, RET inhibitors, and all our patients were already profiled. So we knew that, who would be eligible. And so having that information up front and collecting that information was so useful. And clinical trials and the opportunity to participate in clinical trials is standard of care. Anyway, thanks for those comments. So how? Maybe we can switch gears a little bit and talk about what do we need in terms of what are the minimum clinical, technical, or system conditions that we are required to support effective and sustainable integration of CGP into routine clinical practice. And Dr. Manna already was talking a little bit about things that are happening in Saskatchewan. We heard from Dr. Karam Chandani about Quebec and from his pathology perspective. And what are some of the key barriers? We've already started to hear some of the barriers, things like reimbursement and things like that. But maybe let's talk about what's required, what are some of the barriers, and maybe we haven't heard from you, Dr. Snow, yet. So maybe I'll go to you first, and then come back to some of our other panelists.
Sure. Thank you, Monika. And thank you, Cran, for again involving me today as part of our 4th annual biomarker conference, which I understand is the best one yet. So from my perspective, I'm a medical oncologist in Halifax, Nova Scotia, and so I know the maritime perspective, the Atlantic perspective. And I treat lung cancers and GI cancers. And so lung cancer, especially non-small cell lung cancer, has probably been the solid tumor that's been the most advanced in terms of having detectable molecular targets and therapies that go along with those. And there's no non-small cell lung cancer patient in this country who does not have access to molecular testing. We just don't need it to guide the therapy for our metastatic patients, which is life-changing. Jason, I'm thrilled to hear that were invigorated by that data that I shared. That was some data from the CROWN trial, which was first-line lorlatinib in patients with an ALK fusion. And at the 7-year follow-up point, more than 50% of patients remained progression-free, which is absolutely astounding for this cancer associated with never tobacco users and often a very poor prognosis, very aggressive cancer. So really changing the landscape. But we also need this information now to determine the best treatment in curative intent. Our new standard of care for patients before going for surgery is to do chemotherapy and immunotherapy unless you have a molecular target, including an EGFR or an ALK mutation. And so the question of standard of care really comes down in many ways to access, because while every Canadian non-small cell lung cancer patient has access in terms of funding, the thing is, what does access actually mean? And access comes down to having that information available at the right time to get onto the right treatment. And we know that in our provinces, in our country, there's huge disparity, largely based on geography. If you live in a rural, let alone a remote area, it can be much more challenging even to get that initial biopsy. If your center has to send for molecular testing outside of the hospital, then how does that information come back to a medical oncologist? We know that sometimes the report is there, but if it's not written in a way that's clearly understood by a medical oncologist, especially a community medical oncologist, they may miss a driver mutation that's incredibly important and targetable. And we know that also having that information, as Dr. Auer has mentioned, can lead us to send patients to get something maybe on a trial. And I really think that's part of the standard of care. This has been a very busy month, as Jason mentioned. I was also at another meeting, and at the same time as this one, I'm at an early stage lung cancer meeting here in Montreal called Elixir. And I heard an amazing story yesterday of a young woman who had never used tobacco, who was diagnosed with a very locally advanced but resectable lung cancer. She was a marathon runner in her 20s, and because she had access to CGP, she was found to have an uncommon mutation, was directed to the type of trial we call an umbrella trial. Umbrella trials will enroll patients of the same type of cancer, but they'll go into different categories based on what their mutation is. And because she was able to receive what we call neoadjuvant or preoperative treatment with the targeted therapy, instead of having to have a pneumonectomy and her entire lung removed, she only had to have a lobectomy, and she's back running marathons and has been cured of her cancer. And so those are the kind of stories that also highlight how important it is to have that accessibility. So also a report that you can read and a report that gets to you in time. Having adequate tissue is incredibly important to allow for those trials moving forward. And I think a lot of this really shows the importance of partnerships and collaboration as we move forward. I was thrilled to be at that meeting in Charlottetown. It was the CPQA meeting. It's the Canadian Pathology Quality Assurance meeting. And we saw some really promising data come out where we actually assess quality assurance samples that are sent to different labs across the country. And we did that for non-small cell lung cancer in 2003. And we saw that in many labs, while they came up with the right answer, it was almost 60 days until that report was ready. And that was just too long. Other ones were having the right answer, but it was written in a way that the medical oncologist who was interpreting it did not pick up that this was the type of mutation that they could actually target. But last year, we repeated that. These quality assurance are just that. Feedback is given to the labs. And of all the labs that participated again in the second round, there was clear improvement. Every lab, the fastest and the slowest, had improved their turnaround time. And we saw that there was improvement in the quality of the reports. And so that's why these kind of collaborations that are working between molecular pathologists and medical oncologists together to say, how can we make things better? How can I help you write a report as a molecular pathologist? How can I help you design that to make sure that all my colleagues understand that? And I think it's also so important to continue to collaborate even beyond that. We have great groups like our patient advocacy groups, but also when we meet with the payers, it's so important for us to understand the entire landscape. So if I'm lucky enough to get the ear of the attention of my Department of Health and Wellness for a few minutes, I can talk about how critically it's important that I have access to these drugs. But by the same token, I have to say it's critically important that my molecular lab have access to more molecular technicians, because that's key for that turnaround time. And if they're not aware that that's part of the puzzle, that it doesn't matter if I have access to the drug if I'm going to get my report too late. And this is also where the type of research that's been commissioned by CRAN, we've heard about it yesterday and continuing to evolve, to really speak about this value in terms of dollars and cents in ways that payers can understand, and that can inspire our industry partners to continue to support us as well. Because when we actually show that it's just a drop in the bucket, the cost of the test, when you think about the entire cost of the healthcare system. That young lady, for her one CGP test, who went onto a trial and got better therapy and better surgery, otherwise the standard of care would have been for her chemotherapy and immunotherapy. Probably wouldn't have worked that well. She was never a tobacco user, but those can be highly expensive and certainly highly toxic therapies as well. Also, if it didn't lead to a cure of her cancer, here's a young woman in her 20s who's no longer going to be able to contribute for the rest of her life. And so truly, when you think of the societal value, we have been able to clearly establish not just value in terms of better quality of life and better patient outcomes, but also in terms of nickel and dimes, which is what we have to have those conversations with our payers. So when we all come together from all our different perspectives and have this united perspective and are able to present that, I really think that's how we'll continue to be able to see strides forward in the future.
Thank you, Stephanie. That's great points, and what an amazing story, right? It's always so good to embed all of these considerations in real patient stories and how this makes a difference, and the societal impact. For all of those cost effectiveness analyses, we're supposed to take a societal perspective, but we all work in our little silos, and so thank you for sharing that. I'm going to go to another panelist, but maybe I'll go to Dr. Karam Chandani to sort of we heard from a medical oncologist perspective about some of the what's required, but maybe from a pathologist perspective, and maybe Jason, can I ask you to maybe talk a little bit about the reporting piece, because I think that's a big piece. Because if I understand things correctly, at least from our pathologist in our lab, we're actually running these panels even for but reporting only select drugs, so selected biomarkers, which is so why and what would it take, and then this reporting piece, because if you get a report that doesn't make a lot of sense, then you might miss important information.
Another fantastic question, Monika. And Stephanie in PEI got to see a picture of what a molecular report that is generated from my lab in Montreal looks like. And for our most limited panel, our targeted and focused brain panel, which is the smallest of our genetic panels, the report is 7 pages long. And in fact, what I would consider the actionable information, as Stephanie saw, was about 2 or 3 lines on the bottom of the page. And so we're at this very interesting point in time where I'm speaking on behalf of my colleagues from across the country. I'm very fortunate to know pathologists from every province in Canada. We have 10 different healthcare systems, if not more, if we include the territories. And even within those healthcare systems, we have fragmented and disparate access to some of these tests. Now, I can be very happy to say that in Quebec, we've made tremendous progress in terms of access to testing, but I don't think we are there yet in terms of the timely access to testing. So I think I'm going to be a little provocative, but I'm a neuropathologist, so I'm comfortable being contrarian. Some people have asked the question when to do CGP, and I think it should be at the time of diagnosis. And I think Dr. Manna's point about the ESR1 testing is a beautiful example. Why would we ever want to place patients on a therapy that if we had done CGP, we would know wouldn't work? Stephanie's example from this young woman in Montreal serves as another excellent example of the need to do CGP testing at the outset. But then we get into some very interesting consequences of that. So we are recently approved a new panel, the TSO 500, the next best thing in more comprehensive panel testing. And it is currently approved for only 2 cancers, for ovarian and prostate cancer. Now, I've practiced long enough to know that that's where we start. And I have no doubt that within 3 years, and when I hopefully, if I'm ever allowed to come back, and I'll update you guys, that we'll have more indications for this comprehensive panel. But it brings this unique challenge. If a small panel generates 7 pages of text, the amount of data that we're generating is growing exponentially. And we have the additional complication about what therapies we have access to today is not going to be the same as the number of therapies we have access to in 2 years and 5 years. And Dr. Snow's point is extremely well taken. So it is, my opinion, the job of the pathologist to interpret not only the clinical findings, but the morphology, protein-based biomarkers, and CGP, and deliver a report that is interpretable for the people who ultimately care for those patients. And that can be someone on the surgical side. We're talking about the interesting implications of molecular testing in low-stage colon cancer instead of just high-stage colon cancer, where not every Canadian patient necessarily meets an oncologist. So now we have to communicate to oncologists who are quite conversant in molecular speak, but also some surgeons or gastroenterologists who may be learning about this for the first part in their career. But then the second question, how do we maintain a list of what we have tested for based on the panel that we used at the time in a constantly evolving landscape? And currently, we are simply not powered to do that. So we've already heard about the challenges in the lab. So we don't have enough technicians to do the tests in a timely manner. We have logistic challenge because the systems may not be uniformly organized to ensure timely testing. But we also have a new challenge about how to organize, store, and represent this data in a way that is possibly updatable as my clinical colleagues have access to new therapies. And this is not abstract for me at all. I have diagnosed KRAS mutations in patients where there are active FDA, there are clinical trials right now to find therapies for some of these KRAS mutations, which are not currently targetable. So yet another challenge in terms of how we adapt to the new reality of cancer care in 2026.
Thank you so much. No, as a practicing oncologist, always trying to find all the different, in this shotgun, this testing one gene at a time or one biomarker at a time, and then trying to find different biopsies in different systems. Something is in one EMR, another one is another. Yes, it is a big challenge from a clinician perspective. But so maybe Dr. Banerjee, we haven't heard from you yet in the Manitoba perspective, and I think you're heavily involved in a lot of this work in Manitoba. Can I get your perspective on what you think about how, what are the minimum requirements? How do we do this? Is anyone actually doing this well that we could emulate? So thoughts from you.
Yeah, thank you. So is anyone doing it perfectly? Probably not, but I think we're all heading in the right direction in different ways. I'm a medical oncologist. I practice in two main areas, lung cancer being one of them, and you already heard from Dr. Snow about sort of the value of testing. If we look at our own practice here in Manitoba, we introduced our first CGP assay. It's essentially the equivalent of the foundation assay that Neil brought up at the very beginning. It's the identical thing. It just we can run it in-house. And there is basically no discussion about its value in lung cancer. It is done as part of routine standard of care. The only real limitation we have is if the biopsy material was sufficient to do the assay. And sometimes if that's the case, then we'll go back and get more material. So it's accepted as a standard in lung cancer. The same assay is accepted as a standard in ovarian cancer and CNS tumors, and even in thyroid cancers. And you heard from yourself actually earlier why there's value to doing it in thyroid cancers. And we've approved it in that context because in each of those examples, we need information on multiple genes, and it just makes sense to run a comprehensive assay because it, from a workflow perspective, pretty much costs the same as piecemealing it together as we used to do previously. But it's a challenge to get to this point, and to get here requires many steps. It starts as simple as changing culture of how we approach testing. We spent sort of the first part of this evolution just changing how we communicate between the clinics and the lab, and the standard operating procedures in the lab to ensure that the testing follows a relatively clean pathway. It required heavy investment in infrastructure. We are fortunate in Manitoba that we have a fantastic local foundation that has contributed to that investment. And so we've essentially systematically built around that program. Then it requires validation. Many of the implementation around CGP, it's not that a lab can suddenly today say, we're going to do CGP tomorrow and implement it. It requires a number of very controlled steps to ensure that the assay performs as well in our hands as it theoretically should, and that we meet all the accreditation standards. And then one of the other challenges we run into in terms of implementation is capacity around data analysis and data storage. And this is actually one of the biggest challenges across the country. We had a meeting here in Manitoba where we brought lab leaders from across the country to have a discussion around implementation of CGP, and that was identified as a major issue in the sense that every jurisdiction is really suffering from the ability to keep up from a data perspective. And so implementation can be done, but many very critical steps and resources have to be built at the front end and at the back end to make it happen. Right now, we are only doing 600 CGP cases a year. That's partly limited by the diseases where we're approved to do it. That's also limited by our capacity. We have one instrument that will do this testing. We're in the process of trying to purchase another one. And so there are all these elements that have to really align and be put in place. And then finally, what I haven't touched on, but others have, is really funding, and we have to ensure that the funding aligns all along the way. One of our challenges that we often run into is our diagnostics is performed by one part of our system, whereas this informs cancer care, which is funded through a completely different budget. And often those don't tend to align, and often changes in the diagnostic budget are really only tied to changes in the drug budget. And even though, as Dr. Snow pointed out earlier, diagnostics is just such a fraction of the drug budget, it can often be very challenging to make sure all these elements are aligned and harmonized and moving forward.
Thanks. That's a great description of a lot of the different pieces that need to be in place. And, as we're hearing from all of you from across the country, the variability in how the different provinces are approaching this, and some of the inequity between different types of cancers, right? So, if you're diagnosed with a cancer where this is covered, you'll get access, but some of the cancers, for example, some of the rare cancers where actually this might have some unique benefits, it might be more challenging. Maybe I'll ask Dr. Auer, because Huge, did you want to go before I go to Rebecca? So maybe, and I think we can just open and I have some more questions, but we can go off script a little bit as the conversation evolves, and some great feedback. So go ahead, Huge.
Thank you. Sorry to interrupt. I thought it might be timely to sort of compare the Canadian landscape to the American landscape. We haven't done that yet. And, we always describe the Canadian landscape as a single payer system, but it's really like at least 10 different payer systems. And this is a major barrier. And in the US, we think of it as like a 500 payer system, but people do forget that those who are over 65 are eligible for Medicare in the US. And it has been almost 10 years since Medicare has funded CGP for patients with advanced cancer in the US. So all patients with Medicare who have advanced cancer are eligible for CGP without any out-of-pocket expenses. So that could be Foundation One, but it can be other platforms. So when you compare the US to the Canadian landscape, they have been ahead of us for close to a decade, which is, I think, informative. So we have a lot of lessons to learn. The other thing that they have that we do not have is the NCCN guidelines, which also recommends CGP for advanced cancer patients. And we don't have broad Canadian-based guidelines. I think those two areas are very important to highlight. So apologies to interrupt that.
No problem, Huge. That's a great example and great comments. And actually, we don't really have we have now a good pan-Canadian mechanism for some of the drug approvals, but we don't have anything similar for how we approach the diagnostic piece, which is obviously so important, essential to actually make treatment decisions for patients. So I'll come back to this after. How can we work better together? What do we need to bring the clinicians, the payers, all of that, the partnerships? I think Dr. Snow mentioned, someone mentioned earlier, I think that partnerships and collaboration. I think, Stephanie, that was you in your remarks, how important that is. But so who are the relevant stakeholders that need to be at the table to help move this forward? And obviously, CRAN has been essential in trying to support this conversation, generating data, bringing people together. So thank you, Philomena and the CRAN team. But Dr. Auer, so I'm going to come back to you, and maybe before we move on to the last question, but still come back from your perspective. You talked about the importance of CGP, especially in like things like rare cancers, and obviously have been thinking about, what's comprehensive, what's standard of care. What have you thought in terms of what are the conditions that are needed to make this a reality? If we were to agree what's comprehensive and what's standard of care, what do we need to make this happen?
Yeah, no, that's an excellent question. And sometimes I feel like once we start this conversation, it feels a bit like death by a thousand pins. There's like a thousand reasons why this is complicated and challenging. And I really liked what Stephanie was talking about in terms of, creating these communities of practice around, tackling these challenges together. I think honestly, if we could have all the provinces, federal government all agree that this is something we need to, we need to lead in, we need to change, we need to fix. I do think we as a group of individuals, which includes patients, clinicians, pathologists, radiologists, bioinformaticians, all the way through the entire pipeline, we are absolutely dedicated and creative enough to find pathways for solutions. Reflecting on the disparities across provinces, we had one young patient with cholangiocarcinoma who couldn't get any genomic testing or any biomarker testing at all for cholangiocarcinoma in her province. And in Quebec, they had a pathway to do the testing. And she raised the point that if there was a treatment available in one province and you couldn't get it in my province, I would be allowed to travel to that other province to get the treatment. That is, we have those agreements, those interprovincial agreements. Why can't my tumor travel to another province if we think it's standard of care? And I thought it's such a good point, right? Like, why is it that we accept disparities in this space, which is so critically important, but we wouldn't accept those disparities for a surgical treatment or a radiation treatment, for example? So I do think to Hugh's point, if we had Canadian standards and provinces agreed to meet them, then we could solve a lot of these challenges. And they are challenges, and there'll be creative solutions in each province and probably in each center. But I have no doubt we can work together. Some of the barriers that we think about, like, well, how can we educate all the oncologists to sort of be able to interpret these reports and understand how to work forward. But I have no doubt that if we provide that information, we'll figure out how to do this through multidisciplinary biomarker rounds and not even national platforms, provincial platforms. Like, we are an incredibly tenacious, creative group of individuals. We need the commitment, and I know it has to come from provinces, but maybe it has to come from the federal government saying this is something we need to harmonize across the provinces, and we need to meet this minimum standard. But I'm unfortunately not a politician and don't totally understand how those decisions get made. But I think, Hugh, I had no idea we were 10 years behind Medicare. I wonder if that's something we should be shouting from the rooftops a bit more loudly. Basically Canadians have care that is worse than an uninsured American. That's a problem.
Lots to think about. Obviously, the advocacy piece, and we really, CRAN has really been working in that space. So maybe this is somewhere where they can work with us, the clinicians, and with all of our patient partners to try to help advance and get this on the agenda. And we've already heard most of the labs are already doing this. So it's more about the reporting and expanding. But now we have lots of, so we're going to go off script. Dr. Banerjee, then Dr. Snow, then Dr. Manna.
Thanks. I just wanted to respond to the comment about lack of sort of national effort. And I think it would be wrong for us to not acknowledge some of the efforts that are taking place. So One, and we can debate how effective they are or what how much change they will bring into place, but there are efforts taking place. So one example is through CDA. CDA traditionally has been primarily focused on drug value and costing assessment, value and efficacy assessment, and they have now created a biomarker framework to essentially assess diagnostics. And I hope one day CGP will be part of, or will be one of the approaches that are essentially addressed through that framework. There is also a separate initiative led by several folks, including Dr. Feilotter in Ontario, to essentially bring together a laboratory community of practice. So essentially harmonize what is being done in laboratories across various jurisdictions in Canada, with the idea that if we can have a better idea of what's happening across the country, then we will maybe have fewer pockets of differences that currently exist. So I just wanted to just make a point that there are national efforts, and we need to support those as best we can.
Thank you very much, Shantanu, for raising awareness about those efforts. Is there something that we could be doing better to help support those efforts? I don't.
Well, I know that many people on this this seminar and members of CCRAN have been part of those efforts. So I think a lot of it is continuing the conversations and continuing advocacy within those groups. I think that in terms of the biomarker framework, there's a couple of files going through right now. They're simpler in scope, I think, and it's more to test the methodology and ensure that the process works as intended before we dive into more complex discussions like CGP.
Thanks. Dr. Snow.
I wanted to talk a little bit about molecular oncology boards, where we actually talk about tumors from the basis of their molecular makeup as opposed to, breast rounds or lung rounds. And I think that last year we had a great example from the country of Australia, where they accessed their CGP through funding from a non-for-profit organization. But not just do they get the CGP, but they have access to a national expert-driven molecular oncology board. And they actually interpret that information in a way that's usable by the end user, the clinician, and also say, there's this trial and there's that trial. And I think in Canada, that could be so useful. I consider myself, I'm an academic oncologist. I keep on top of the literature. And I'm also very privileged to be part of informal networks within my tumor types, where I could just send an email to Princess Margaret and say, do you guys have anything for MET amplification after progression on a first line, TKI for EGFR, those types of things. But not everybody has access to that. And especially community docs, I think, really don't necessarily feel comfortable coming forward, be like, I don't know what this mutation means, or I don't know what to do with it. There can be sometimes embarrassment, and nobody can keep up with all of this. And I really think that it's so important that you have those relationships, again, in those partnerships. Sometimes I see something on the molecular report, I'm like, I don't know what that is. So what do? I pick up the phone and call Mike Carter, my molecular pathologist. I'm like, is this what I think it is? Is this a HER2 in like exon 20 insertion? He's like, oh yeah, that's what it's like. Well, can you write it like that so I can actually recognize it? Because I do have a drug for that one, or at least a trial. But if we had access at a virtual molecular oncology board, I think that would be so helpful. And it would be another great way to bring the stakeholders together. We do have some wonderful molecular oncology boards already in Canada. Vancouver is an excellent example of that. And their group is really pushing a lot for truly comprehensive genomic profiling with full exome and transcriptome, detailed delineation. They're way ahead of the game in some of their research. But just basic things like that, I could really think move the bar and make sure that that information is being interpreted. And if we have a process, then that gets over all the little skips, because you're not going to have a molecular oncology board for every single tumor type at every single hospital.
Thanks, Stephanie. That's a good point. Obviously, this information is becoming more complex. New things that were not actionable are becoming actionable. The reports are, very complicated. Someone's saying 7 pages, but really 2 lines are really the important ones. So mechanisms, and we are in 2026, what's the role of AI to help us interpret all of these things? And also for patients, right? Patients are getting results of these copies of these results. How do we help support them? If we're struggling with trying to keep up with everything. So maybe we'll come back and talk a little bit about that. But before, I'm going to go to Dr. Emenegger. Urban?
Yeah, I have 2 comments based on my experience as a actually prescriber of these testing. So the first thing is that we haven't defined what comprehensive is, but for instance, in our case, and this is I think Ontario-wide the case, we get 4 gene results reported, but the panel actually that they use is 160 genes. And the reason why I don't get all the information of the 160 gene panel is that it's a lack of manpower. And I can't imagine that in the time of AI, there's no easy way actually, or some reasonable way to make this information available. This information sits somewhere, but it doesn't become available to the clinician. And it might not be always actionable mutations, but if I, for instance, know my patients that they have a p53 mutation, or they have a RB1 deletion, or they have a p10 mutation, this has an impact on how I will follow these patients, but I'm more concerned that they will not do so well. So that's the first comment. The second comment is that, and again, it's about comprehensive. We know in prostate cancer that prostate cancer amongst the cancer types with the highest rate of heritability, more than 50% of the risk of prostate cancer has to do with genetics. And this also means that in prostate cancer, we do both in parallel. We do germline testing and we do somatic testing. Somatic testing is primarily for the patient. Germline testing is for the families, which it can result in cascade testing. And I have seen quite a few patients where my patients actually tested positive, let's say, for a BRCA2 defect. And then we actually do germline testing, realize that it's actually a BRCA2 defect that runs in a family. And I have now sons of my patients that know that they have a high risk of having prostate cancer. I have daughters of my patients that realize that they have a high risk of having breast cancer, high risk of having ovarian cancer. And that empowers them actually to do screening earlier. And the way we do it now will be completely different in 10 years down the road. I think we will become better and better in actually helping these family members that have a higher risk of developing cancer to not become cancer patients. And initially, I mentioned to you that I treat metastatic prostate cancer patients, and I cannot cure my patients, but I hopefully can contribute that at least some of the family members do not become cancer patients. And as such, I can help to somehow cure them as well as a proxy of what we do with our patients.
Thank you so much, Ruben, for highlighting that other use case of some of these test results, not just for the individual affected by cancer, but by also for their family members. And although that does add some more complexity in terms of managing all of these results and in this evolving field. Dr. Manna.
Thank you. So I also wanted to add on to some of the previous comments that were raised. I think you mentioned what can we do better? How can we sort of engage our stakeholders and make these changes happen? I think I just wanted to comment that just platforms like this allow us to, bring awareness, understand what other centers and other provinces are doing so that we can collaborate and partner together to help make change. Otherwise, if we're just working in our separate silos, we don't necessarily know what's going on, and we can't necessarily form these connections. So I think this is very important, first of all. Second of all, and just what Dr. Banerjee mentioned, I think all of us are recognizing the vast changes that are happening, what's happening down to the south, things happen a lot faster in comparison. And so I think it's also important to recognize that, it's very challenging to have national guidelines, but when they do come together, it can really help impact change. And I've been fortunate to be part of one of our breast cancer coalitions. So it's called the Real Alliance. And we've actually painstakingly put together national Canadian recommendations in terms of management of breast cancer. And we've seen that it's helped sort of impact policy changes, impact clinician management and guidance when it comes to treating breast cancer, and help overturn some negative recommendations for implementing certain therapies. For example, pertuzumab for HER2-positive early-stage breast cancer. Initially, that was a negative recommendation, and now, it's been overturned, and we're just waiting for reimbursement. But in addition, we're not just implementing recommendations and guidelines for therapies, but also biomarker testing and even staging and imaging, for example. And so with that, there's also another group where we're trying to put together guidance for biomarker testing. And as we mentioned, we have a fragmented, country with various different systems. So if one center were to implement CGP, like we mentioned in BC, they're already doing, an exceptional job. Instead of reinventing the wheel at each center, how can we learn from that one center and essentially start it up in all the other hospitals and labs across the country? So that way we're not having to start from scratch. So these are some of the implementations or the work that's being done sort of to try and ensure that we can get CGP and all these other testing up and running across the country, and how we can all collaborate together so we're not, reinventing the wheel every single time. So I think it's important, once again, to know that there's a lot of work being done, but of course it does take time. And I think the biggest factor is also funding, right? So we need to engage not only government, but we also have our industry partners as well, who also have big stakeholder, in this as well, because if we are able to at least get biomarker testing done, we have therapies that we can also use for our patients. So I think engaging everyone in these initiatives can really help with the testing overall.
Thank you so much, Mita. That was great comments. So we have about 15 minutes. I'm going to go to sort of maybe get everybody's thinking caps on. Maybe we'll do a brief, what's the next, what's one actionable thing we could do as the next step to help. I have some ideas, but I just so get your thinking caps. And while we're doing that, I'm going to see if there's any questions in the audience. So I'm just so from the session yesterday, it sounds like there were multiple panels. So I'm going to take one or two questions, but in the meantime, think about this question: what is the one actionable thing we could do as the next step to help move this forward as a recommendation that CGP should be the comprehensive CGP should be the standard of care? So the question is from. It sounds like even when multiple panels are tested, non-actionable findings are sometimes not mentioned in the report. So we talked about this, right? Given the evolving landscape of targeted therapies from a clinician's perspective, do you think having a full non-pre-filtered report, including currently non-actionable findings, would be helpful? I mean, anyone, feel free to put up your hand. I mean, as I indicated earlier, as someone who's used to treat thyroid cancer when there were no therapies, and doing this, I just got a list of genes. Eventually it became useful. Just having to access it in a database was the key thing for me. But maybe Shantanu, then Stephanie? What do you think?
From my perspective as someone on the clinical side and on the lab side, I would say that I see no reason why everything shouldn't be reported. I think we already have rules about how to classify them, and as long as we're essentially following those rules, I think it's important to have that information out there for the reasons you've explained. I think one of the challenges in some jurisdictions, and I believe Ontario is one of them, is that my understanding is that there's actually some legal or accreditation reasons that non-funded or non-approved biomarkers can't be reported. And so that's not a technical issue, nor is it a clinical necessity issue. It is an administrative issue.
Great point. And maybe I'm going to go to Stephanie and then Rebecca, but actually I was going to say, Jason, I don't know, as a pathologist, if you have any more insight into that particular piece, because that's what I keep asking our pathologists. Why can't you just report it and we'll worry about trying to figure out if it's actionable? Stephanie.
My perspective is almost purely clinical. I definitely think that we should be given this information. Sometimes a patient is diagnosed and they're following with you for years and years and years, and our clinical landscape changes so quickly. And when I do my note, I have a little synopsis up top of what the diagnosis and the treatments have been on. And if there's any kind of mutation or a biomarker that I'm aware of, I put it there and it gets cut and paste every single time. And sometimes I was putting in something like 10 years ago, a HER2 mutation in a lung cancer patient, which didn't mean anything. But today it means something. Today it means that we've got drugs like zongertinib and TDXD that actually can work in that patient. If I hadn't been cutting and pasting for 8 years, I probably would have forgotten. And so that's why there are ways to report this. There's the tiers. Tier 1 is immediately actionable, very important, the different tiers. And we even have variants of uncertain significance. And 8 years ago, 10 years ago, a HER2 insertion mutation in a lung cancer patient with a completely variant of unknown significance. But today it's different. At the very least, I argue that you should tell us what you're testing, because sometimes you don't know if there's a certain test that's on the panel or not. So our current panel, for instance, doesn't have NRG fusions. And there's a particular type of lung cancer, if it's a mucinous histology and it doesn't have a KRAS mutation, the pretest probability of having that NRG fusion goes up really high, and we have drugs for that. But I know, because I've had to ask, it's not on my panel, so we cooperate. I get that tissue sent to Moncton, where their supersized panel does have that. And so we can actually do that. So at the very least, if we know what's been tested and not tested, and again, I might come back from World Lung in September, having gone to, a phase 1 kind of report session, like, oh my gosh, we have a brand new biomarker or a brand new drug that seems to have actionability. And guess what? They're opening up that trial in Princess Margaret. So if I can go back and see if my patient had it, again, I can make those concerted efforts to get them to those trials. So I, from a clinical perspective, think it's amazing just to let us know it's out there, but make it clear if it's not a tier 1 at this point.
Thanks, Stephanie. Information is power. Rebecca.
Yeah, I would just echo what has already been said. It's about future proofing for what we don't know and what we know could be on the horizon. And I think it's also would be very valuable for drug approvals like CDA. So in cholangiocarcinoma, there was a really challenging CDA decision around pemigatinib for FGF2 translocations. And one of the comments that they made was that we don't know for sure, because it was not a randomized study, rare cancer, if FGF2 translocations just mean a better prognosis. And it wasn't the drug at all. It's just that you've subselected a group of patients with a better prognosis. Well, if you had all that information already, it would be very easy to go back and say, before this drug was available, or when it was only available in clinical trials, those that had an FGF2, this was what their prognosis looked like, So I think there are a lot of places where for that individual patient, but also for making decisions, health economic decisions, approval decisions for funding could be supported by understanding what is the natural history for that biomarker in the absence or presence of a particular drug. So I completely echo that we need to be forward thinking in the report.
Thank you. Jason.
So at first, I'd like to thank Dr. Snow, because back in Prince Edward Island, I asked her a question that's similar to what we're talking about. Are you, as a clinician, comfortable with me reporting things that may not be currently actionable, but that may be actionable in the future? And I'll give both an easy and a complicated example. So just old-fashioned immunohistochemistry for HER2. So I've added this to all solid tumor metastases for several years. The reality is we don't even have consensus on the scoring system for how to interpret HER2 in different primary sites of cancer origin. So I've been just doing this perfectly descriptively. So I describe the clone, I describe the pattern, the number of cells, and the intensity, understanding that the classification or the interpretation guides are evolving rapidly. That information may not be usable now, but it is my hope that if ever my oncologist needs that information, that they're able to access that in a timely way. Conversely, going back to CGP, I heard a story in Vancouver 3 weeks ago Philomena presented at our conference on biomarker and precision therapy. And there are genetic panels that not only test genes that are relevant for cancer, but responses to different drugs, so pharmacogenetics. And we have to remind ourselves, and I think I mentioned this earlier, that the genetic results are not just going to oncologists, but increasingly they're going to go to different allied healthcare professionals and even patients. So the pharmacologists in British Columbia, even though they had access to this information, were not yet comfortable, or they didn't feel confident to receive those results with a possible medical legal implication for their practice. So we have an interesting complexity here that we are generating data, but not everyone who's getting this data is necessarily comfortable or even necessarily trained to be competent to interpret some of these results. So that's a challenge. Secondly, we talked about my honest confession that our reports are not stored in a way that I consider particularly useful. We issue what is a PDF. It is a paper report. And so we list all of the genes that we test there, but it's a dirty secret. Just listing the genes that you test is not nearly enough information, certainly for panel-based testing. But even though we do exome and transcriptome, while we report the number of genes that we evaluated, I'm not telling you about the copy number. I'm not telling you about our depth of coverage or other potential pieces of information which could also be incredibly useful. And we know cancers are heterogeneous. We know sampling is an issue. We know tumor cellularity is an issue. So even reporting the results, reporting them all and putting them in a piece of paper is still insufficient to provide my colleagues with the information they may need in the future. So finally, we talked about one actionable thing. And again, Stephanie brought this up, so I'm merely repeating what I think is an excellent idea for Canada, and I do think it's actionable. I think if we standardize how we report our results so that at least as an oncologist, you're not trying to interpret a result depending on which hospital you sent a test to or which province you live in, to me, that is one of the lowest hanging fruits, and at least I believe has a concrete potential to improve patient care, particularly in community centers where patients are receiving these results for only a handful of patients over the course of their career.
Thank you so much. So I heard one idea, standardize the reports. Neil, I was going to come back to you. So floor is yours. And then I'm going to, if we have time, or if people want to put something in the chat, I don't know if we can use the chat to get some of those ideas, then please do. Neil.
Thank you. First of all, I want to say just from listening to all this, how tremendously encouraged I am. If we can go back 6 years, I discovered Foundation 1 in genetic profiling online myself and raised this with the oncologist and was discouraged from doing that at that time. Think where we've come from. And when Jason said that we should be doing this at time of diagnosis, I yelled out, yes, we should. And if I can just go back to what I touched on in my opening statement, it's the psychology aspect I'd like to bring. Contrast an experience like that with what my wife was driving her grandchildren to school and was told on the phone that she had probably a terminal cancer. Imagine if we could now just go into the doctor's office and say, yes, we have this diagnosis. However, we're going to do comprehensive genetic profiling. There are tremendous strides being made, and hopefully we can get something in terms of treatment. So I'm really encouraged by what I'm hearing in that way. And I would just say to all the patients who are on this, you're already brave because you're taking part in your own care by being in this conference. Extend that bravery when you go into your doctor's office and advocate for yourself. Yeah, so I just want to say thank you to everyone for all their participation in this. I'm very much encouraged.
Thank you so much, Neil, for your perspective. And indeed, I think the patients are important partners, and their caregivers are such important partners in this journey. This is it's really things go better when we work together. And this has been really a great example when you bring perspectives from across the country and from different specialties around the table, just really show you what the value is of working together in those partnerships and collaborations. And Jason, there was a question, one of the questions, we won't have time to get to it. What's the role of the pathologist? Well, after, I think we heard this from multiple panelists today. We're all talking to our pathologists. Maybe you should be coming to clinic and talking with our patients, because given the complexity of these diagnostics, right? Like it's very hard to keep up. Even for academic clinicians, never mind for our busy community partners, and then for our patients. So anyway, I think we're almost out of time. I don't know, Philomena or Cassandra, if you wanted to have any last minute comments. I've heard we have to hold our politicians accountable. We have some there are some consensus guidelines that I think would be helpful around standards for reporting. I think Rebecca was talking about, maybe we just need to all agree of what do we mean by the minimum sort of, what are the minimum standard that we should be meeting? So, and the patients are our strongest allies. So thank you very much, everyone, for your perspectives. I've learned a few things, and really nice to sort of hear the conversation, and that we're, I think, all on the same page. It's just how do we move this forward? So Cassandra or Philomena, I don't know who's gonna.
Yeah, thank you so much, Monika. Wow, what such rich expertise in this discussion. There is so much work to be done, and CRAN is very grateful to be able to work with thought leaders like the experts on this panel today to be able to advocate for health system transformation on behalf of cancer patients right across Canada. So thank you to each and every one of our panelists for this really robust discussion. From this discussion, we've heard that CGP is no longer an aspiration, but it is a reality, and there is urgency for equitable access across Canada and across tumor types. There's urgency also in challenging the status quo with respect to what is the standard of care and the kind of more broad understanding of what comprehensive testing really entails, which is certainly a moving target in this evolution of precision oncology that we are living in. And an ongoing challenge as well is to keep pace with the rapid advancements in the field and the data storage requirements that come along with that, so that our Canadian cancer patients are receiving a high quality of healthcare. So thank you again for this wonderful, wonderful discussion, which we look forward to capturing in our manuscript.